Welcome back to Signal 2030, where we explore the scientific breakthroughs that are shaping our future.
Today, we're tackling something huge. A question that honestly sounds like it's straight out of science fiction, but it's becoming science fact pretty rapidly.
Yeah, what if everything we thought we knew about aging, the whole process, was actually wrong?
What if it wasn't this inevitable slide into decay, this one-way street, but more like a software problem, something you could potentially debug?
It's a mind-bending idea, and that's exactly what we're going to unpack today. We're doing a deep dive into Harvard geneticist Dr. David Sinclair's information theory of aging.
Right. Our mission today is to really get into this bold theory. We'll look at the science behind it, the foundations.
We'll check out the pretty groundbreaking evidence coming out of the labs. We're talking primary research reports here.
And then how is this translating, into actual potential therapies that could completely change what it means to grow old?
So let's dive right in. Forever, we've sort of accepted aging as just inevitable wear and tear, right?
Yeah, the traditional view is a hardware problem.
Yeah.
Your body just accumulates damage over time.
Things like mutations popping up in your DNA, or those little caps on our chromosomes, the telomeres getting shorter.
Or just cellular junk building up. And the best you could hope for was, well, maybe slowing it down a bit. Damage control, basically.
But this information theory of aging, the ITOA, which David Sinclair really champions, it flips that script completely.
It really does.
Yeah.
It says aging isn't primarily a hardware problem. It's not mainly about your DNA getting irreversibly damaged.
Instead, it's a software problem.
Exactly. It's the progressive loss of epigenetic information. That's the system that reads and, importantly, regulates your genetic code.
Okay, unpack that epigenetic part a bit. The hardware-software analogy helps here, I think.
It's a great analogy. Your DNA, your genome, that's the hardware. Think of it like the digital code on a computer disk. It's remarkably stable throughout your life, actually.
Right. But the epigenome, that's the software.
Precisely. It's this complex layer of chemical tags and proteins that wrap around your DNA. It tells the hardware, the DNA, which programs to run, which genes to turn on or off in any given cell.
So a liver cell knows it's a liver cell, a brain cell knows it's a brain cell, even though they have the same DNA hardware.
Exactly right. That's the epigenome at work. It provides cellular identity.
So aging, in this new view, is like that software getting corrupted, getting noisy over time.
That's the core idea. Like scratches on a CD, maybe.
Right.
The underlying genetic information, the song, is still there. But the cellular machinery, because of this epigenetic noise, can't read it correctly anymore.
And that leads to cells forgetting what they're supposed to be doing.
Yeah, they lose their identity. They might turn on the wrong genes. This dysfunction contributes massively to the diseases and frailty we associate with getting older. It's not a loss of the instructions. It's a loss of the ability to interpret those instructions.
Okay, and this is where it gets really radical. If aging is information loss,
then maybe that information could be restored. Sinclair's own words are something like, if my information theory of aging is correct, you can actually reset that.
Wow. So we're talking potential age reversal, not just slowing things down, but actively, like, rebooting the system,
Yeah.
polishing the CD.
That's the promise.
Yeah.
It fundamentally shifts the goalposts. Now, to really get this, we need to understand that epigenome, that software, a bit better.
Right, the conductor of the cellular orchestra.
Nice.
Yeah.
Yeah, it uses things like chemical tags, methylation is a big one, and how tightly the DNA is wound around proteins called histones to control which genes are active.
But the ITOA says this precise control erodes with age, this epigenetic drift.
Exactly. Patterns get scrambled. Tightly wound DNA, which should be silent, might loosen up. Genes that should be off might flicker on. Cells get confused.
They lose their identity, become less efficient, and that fuels age-related problems.
Right. And here's a key insight from the theory. The major source of this disruptive noise isn't just random decay. It's actually tied to something essential for survival: DNA repair. Our DNA gets damaged constantly, thousands of breaks every single day, from metabolism, from the environment.
Especially serious things like double-strand breaks, where both strands of the DNA helix snap.
Yes. And when that happens, the cell absolutely has to fix it. It sends in the repair crews. And guess who's part of that crew?
Let me guess. Epigenetic regulators.
Bingo. Particularly a family of enzymes called sirtuins. There are 7 of them, SRT1 through 7.
And their day job is managing the epigenome, keeping things tidy, and maintaining cell identity.
That's right. They help keep DNA tightly packaged, genes silenced where they should be. But when there's a major DNA break, especially a double-strand break, sirtuins have to drop everything.
They leave their regular
They
posts.
get recruited to the damaged site to help coordinate the repair. Now, this happens over and over, decade after decade.
And the problem is, they don't always go back to exactly where they started afterwards.
That seems to be a crucial part of it. This constant relocation and return isn't perfect. Little errors, these sort of epigenetic mislocalizations, accumulate over time. That's the noise that corrupts the software.
So wow, the very act of protecting the genetic hardware degrades the epigenetic software. That's quite a paradox.
It is. It seems to be a fundamental trade-off. And there's another layer to this problem involving sirtuins.
Oh.
They need fuel to do their jobs, both the epigenetic maintenance and the DNA repair. That fuel is a critical molecule called NAD+, nicotinamide adenine dinucleotide.
And I've heard about NAD+ levels declining with age.
Dramatically.
Yeah.
By as much as 50% between our youth and middle age.
So if NAD+ levels drop, the sirtuins are starved of fuel.
Exactly. They can't do their epigenetic housekeeping properly, and they can't respond as effectively to DNA damage. It becomes a vicious cycle, accelerating that epigenetic noise.
Okay, so if aging is this gradual corruption of the epigenetic software, driven partly by DNA repair and falling NAD+ state, the obvious next question is, can we fix the software? Can we reboot?
And that brings us to the work of Shinya Yamanaka. His Nobel Prize-winning discovery in 2006 was absolutely pivotal.
The Yamanaka factors, right? Those 4 specific genes.
OCT4, SOX2, KLF4, and CMIC, or CMYC.
He found they could reprogram a fully differentiated adult cell, like a skin cell, all the way back to an embryonic-like state, a pluripotent stem cell, essentially resetting its epigenetic clock to 0.
But reprogramming cells completely like that in a living animal is dangerous, isn't it? Risk of tumors, losing cell identity altogether.
Huge risk. That's not the therapeutic goal. The breakthrough for age reversal came later, with the idea of partial reprogramming.
Meaning?
Expressing these factors,
Maybe just OSK, the first 3 Yamanaka factors, leaving out CMYC.
which is linked to cancer for only short periods, just enough to kind of clean up the epigenetic noise and rewind the clock a bit, but without erasing the cell's identity.
So a liver cell stays a liver cell, but it becomes epigenetically younger.
That's the concept
It's proposed as the way to access that pristine backup copy.
of youthful information Sinclair talks about, wiping away the accumulated errors.
Which leads to this sort of two-pronged approach for longevity, right?
Exactly. On one hand, you have maintenance,
Things you do continuously, your lifestyle.
maybe certain supplements to slow down the accumulation of that epigenetic noise, like regular oil changes for your car.
And on the other hand,
Restoration, periodic, more powerful interventions
Maybe like OSK gene therapy.
to do a system-wide reset, to actively reverse biological age in tissues. That's the engine overhaul. It's a synergistic idea.
okay, this theory is compelling. It hangs together logically, but the proof. Where's the evidence that this actually works, that epigenetic noise causes aging and that resetting it reverses aging?
That's the crucial question. And the lab results are starting to come in, and they are pretty remarkable. A major one was the 2020 paper in Nature from Sinclair's lab.
The vision restoration study in mice.
That's the one, a landmark piece of work. They used a safe virus, an AAV, to deliver the OSK genes directly into the retinal ganglion cells of mice, the nerve cells connecting the eye to the brain.
And the result?
They were genuinely stunning.
They restored lost eyesight in old mice. They halted and even reversed vision loss in a mouse model of glaucoma.
Wow.
Maybe even more strikingly, they significantly promoted nerve regeneration after injury to the optic nerve, something nerves in the central nervous system normally don't do well at all.
And crucially, they looked at the molecular level, at the epigenetics.
Yes, they confirmed that the treatment reversed age-related DNA methylation patterns. This was strong evidence that these epigenetic changes weren't just biomarkers of aging, they were actually driving the dysfunction.
And has this been replicated, moved beyond mice?
It has successfully replicated in non-human primates, which is a critical step towards humans. That work helped pave the way for the human trials we'll talk about.
Okay, so that 2020 paper showed age reversal was possible in a specific context, but there is still that deeper question. Is epigenetic information loss really a fundamental cause of aging?
Right, correlation versus causation. To tackle that, Sinclair's lab undertook another, really ambitious study published in Cell in 2023. They created what they called the ICE mouse,
ICE.
inducible changes to the epigenome. These mice were genetically engineered so the researchers could induce specific types of DNA breaks whenever they wanted, but without causing mutations. The genetic hardware stayed the same.
Ah, so they could trigger the DNA repair process repeatedly and test the hypothesis that this process itself, by disrupting the epigenome, would drive aging.
Precisely. In the first part, they induced these breaks in young mice, and they saw accelerated aging. The mice got frail, gray, their tissues aged faster physiologically, cognitively, and by molecular measures like the epigenetic clock.
So they drove aging forward seemingly just by scrambling the epigenome through repair.
That was the interpretation presented. Strong evidence that epigenetic information loss causes aging. Then they did the reverse.
They treated these prematurely aged ICE mice.
Yes, with the OSK gene therapy. And they reported a remarkable reversal of those aging phenotypes. Sinclair described it as rebooting a malfunctioning computer.
Being able to drive aging forwards and backwards like that sounds like powerful support for the information theory.
It was presented as such. Now, we have to be clear, science thrives on scrutiny. And this high-profile Cell paper definitely received some.
There was some debate, right? Critiques.
Yes, vigorous debate. Some prominent researchers, like Charles Brenner, argued strongly that the enzyme used to make the DNA breaks, IPIOI, might itself be toxic to cells.
Cytotoxic, meaning it kills cells.
Right. Their critique suggested the accelerated aging seen might be due to widespread cell death and inflammation, not purely the result of epigenetic noise from the repair process itself. They raised concerns about controls and interpretations.
How did Sinclair's lab respond?
They published a formal rebuttal defending their methods and data. It's a complex scientific discussion ongoing, really.
So what's the takeaway for us listening?
I think it highlights how rigorous science works. Big, bold claims require extraordinary evidence and face intense scrutiny. Proving a grand unifying theory of aging is incredibly challenging. But the debate itself pushes the field forward.
Absolutely. Okay, so moving from the lab bench towards the clinic, how is this theory actually being translated into therapies that could help people?
This is where companies like Life Biosciences come in, which Sinclair co-founded. They're really focused on taking these ITOA principles and turning them into clinical treatments.
And their lead program is based on that vision restoration work.
Exactly. It's a direct translation of the OSK gene therapy used in the mice and primates, using a safe AAV virus to deliver the genes.
Delivered into the eye, presumably?
Yes, via injection into the eye. And a really key feature is that the gene expression is inducible. You can turn it on and off using a common antibiotic, doxycycline.
Ah, so you get precise control over the reprogramming process. That sounds important for safety.
Crucial. They're initially targeting tough, age-related eye diseases with few options,
like NAION, sort of a stroke in the eye, and glaucoma, a leading cause of blindness.
And the timeline. When might we see human trials?
They're aiming to start human trials in the second half of 2025. It's a really exciting prospect.
It really is. But you mentioned gene therapies.
Mm-hmm.
They tend to be incredibly expensive, right? Hundreds of thousands, even millions per treatment. How do we get past that potential barrier for accessibility?
That is a massive challenge, and it's where another technology comes into play in a big way. Artificial intelligence, AI.
How is AI helping here?
Sinclair says AI is now central to everything his lab does. The major goal is to find alternatives to expensive gene therapy. Specifically, they're looking for cheap, simple chemical cocktails, essentially, that can mimic the rejuvenating effects of OSK.
And AI speeds up that search dramatically.
Massively. They have algorithms that can analyze microscope images of cells and determine their biological age almost instantly. This allows for super rapid screening of thousands to millions of chemicals.
Wow, and they can model things virtually, too.
Yes, they've identified key epigenetic enzymes, like levers for reversing aging. AI can then virtually screen trillions of potential drug molecules against these targets simultaneously. Work that used to take decades and billions now takes months.
Have they found anything promising yet with this AI approach?
They have. Sinclair has mentioned finding a chemical cocktail that, when given orally to old mice for just 4 weeks, made them physiologically and biologically younger.
Taken orally, like a pill. That would be a game changer for accessibility and cost.
Absolutely. It shifts the strategy from hardware, adding genes to software, using small molecules to send the right commands. The aim is to make age reversal potentially as simple and affordable as taking an aspirin one day.
That's incredible. And this whole field of epigenetic reprogramming, it's not just Sinclair's lab anymore, is it? It sounds like it's exploded.
Oh, completely. It's ignited this incredibly competitive and, perhaps more importantly, extremely well-funded industry. It's a new race.
Who are the big players?
Well, there's Altos Labs, launched with something like $3 billion from backers, including Jeff Bezos. They have a scientific dream team, including Yamanaka himself. They seem focused on really deep, fundamental biology of cell rejuvenation.
$3 billion. That's serious investment.
Then you have New Limit, founded by the CEO of Coinbase, Brian Armstrong. They're very focused on using AI and machine learning to discover new combinations of factors to rejuvenate specific cell types, targeting diseases like liver disease or autoimmunity.
And Life Biosciences, Sinclair's company, focusing initially on the eye gene therapy.
Right. So the point is, age reversal, particularly through epigenetic reprogramming, is no longer fringe science. It's a major biotech venture now, driven by this core belief that aging is programmable and ultimately solvable.
So looking ahead, what does the future realistically hold? Sinclair makes some bold predictions, 2035 for effective therapies, possibly pills.
He does. And this ties into the concept of longevity escape velocity, or LEV.
Explain that again.
It's the idea of a future point in time where, for every year you live, science advances enough to extend your remaining lifespan by more than a year. So you effectively start outrunning death, or at least age-related death.
And Sinclair thinks this is feasible.
He believes it's feasible, yes.
Huh.
He's talked about the possibility within our lifetimes of being able to reset yourself multiple times, maybe 20 times, maybe even 100.
Which leads to his famous line, the first person to live to 150 has already been born.
Right. Likely someone who's a teenager today, he suggests. It does make you wonder, were we born just one generation too early for all this?
It's a thought, and his advice then is?
Stay healthy now. Do whatever you can with current knowledge, diet, exercise, managing stress to increase your health span today so you're around to potentially benefit from these breakthroughs when they arrive.
The economic implications alone are just staggering if this works.
Mind-boggling. Estimates suggest extending healthy lifespan in the U.S. by just 1 year could be worth $86 trillion to the economy. 10 years, $367 trillion. Globally, a 1-year increase is maybe $38 trillion.
So investing in longevity research isn't just about health. It could be one of the most powerful economic policies imaginable.
You could argue that, which makes it kind of ironic, Sinclair often points this out, that basic research funding from traditional sources, like government grants for his own lab, can be precarious, while the private sector is pouring billions into the commercialization side. There's a potential disconnect there.
And the equity issue remains. Initial therapies, especially gene therapies, will likely be very expensive.
That's the reality. But Sinclair's stated mission, especially with the AI-driven chemical approach, is to democratize it. To find those cheap oral alternatives and eventually, through patent expiration, make rejuvenation potentially accessible to everyone.
So while we wait for these potential breakthroughs, what about now? You mentioned Sinclair's personal regimen and the maintenance side. What does that involve? Bearing in mind, this isn't medical advice.
Right, definitely not medical advice, just reporting what he shares about his own approach, based on his understanding of the biology. It's built around stressing the body in beneficial ways to activate those natural longevity pathways.
3 pillars, you said.
Yeah. Diet is one. He follows a largely plant-based diet, not much meat, very little alcohol, and incorporates intermittent fasting, often eating just 1 or 2 meals a day.
Okay. Pillar 2?
Exercise. He calls it one of the most potent pro-longevity drugs, a mix of resistance training, lifting weights, and cardio like walking or running.
Makes sense. And the 3rd pillar is supplements.
Yes. Though he emphasizes his stack is dynamic, he adjusts it based on new data and his own biomarkers. But some core components he's often mentioned include NMN, which is an NAD plus precursor.
To boost those sirtuins we talked about.
Exactly. And things like phyzitine and quercetin, which are thought to act as senolytics, helping clear out old senescent cells, and may also activate sirtuins. Plus some others like alpha-lipoic acid.
What about things like metformin or rapamycin that get talked about a lot?
His views seem to have evolved. He's expressed less enthusiasm for taking metformin regularly, preferring berberine perhaps, and seems less keen on rapamycin currently, citing a lack of clear epigenetic age reversal effects in human studies he's seen. He might use them occasionally, but they don't seem core for him now.
Interesting how the science keeps moving. So this whole protocol is about shoring up the body's defenses now.
That's the idea. It's the maintenance strategy, trying to slow down the accumulation of that epigenetic noise while we wait for the potential restoration therapies to arrive.
Okay, so wrapping this up, this deep dive really shows how David Sinclair's information theory reforms aging fundamentally. Not as inevitable decay, but as a potentially reversible loss of information.
It gives us a powerful new lens, a framework for understanding how we might not just slow aging, but actually turn back the biological clock.
And the path forward looks like this dual strategy. Keep up the maintenance lifestyle, maybe targeted supplements.
Combined with, hopefully in the future, periodic, powerful restoration therapies using things like epigenetic reprogramming.
We're clearly on the edge of something profound here. Gene therapies are heading towards human trials. AI is turbocharging drug discovery. The pace is incredible.
Absolutely. Challenges remain, of course, the scientific debates, the cost and equity issues ensuring safety, but the momentum is undeniable.
And the ultimate goal, it seems, isn't just adding years to life, but adding life to years. A longer health span free from disease and frailty.
As Sinclair puts it, being healthy, having connections, why would you want that to end? So, the final thought for you, listening.
Yeah.
What would you do with a continually restored health span if age wasn't a limit in the same way?
How might that change not just your life, but our whole society, maybe even by 2030 or 2035?
That's something to chew on as you reflect on this episode of Signal 2030.
Thanks for joining us.
Bye for now.